In a world where land is our most precious resource, soil erosion looms like a threatening shadow, a shadow that puts our food security, our environmental health, and our economic stability at risk. Hi, my name is Ricardo Howard and I am team leader of iGEM EVOLUTECH Panama, the school team participating in Design League 2023. Our project, GROUNDSAVIOR, has been under construction for over a year, with an interdisciplinary group of young students. Now, we are excited to present it to the world. Soil erosion in our region is a significant challenge, especially in the La Villa River, with notable damage to the soil structure, loss of surface layer, lower fertility, and increased sedimentation. The dynamics of the river are constantly changing, aggravated by extreme weather events linked to climate change, making it more prone to flooding and canalization. The turbidity of the river has increased, impacting the quality and quantity of water supply by the Chitré treatment plant with a greater risk of contamination. The quality of the water in the La Villa River deeply concerns our community, affecting the environment, human and animal well-being. The atrazine spill in 2014 was a crucial event that prompted us to address this problem, as it directly affects a significant portion of the population and caused numerous cases of disease due to the exposure to this chemical. We commit to doing everything we can to mitigate impacts on our community.
Panama, despite being a relatively rich country in the region, especially in Azuero, its wealth is not always correctly distributed. The funds allocated to these conservation efforts are small and scarce for the amount of work and materials that go into these processes. This then affects a series of problems for both the environment and for ourselves as citizens. Soil erosion in Panama is a critical problem that affects biodiversity, food security, and water supply. Deforestation, intensive agriculture, mining, and climate change are some of the main causes of soil degradation in Panama. According to the map of regions threatened by degradation and drought of the Ministry of Environment of Panama, Mi Ambiente, 27% of the Panamanian territory is degraded or threatened with drought. This problem extends to other regions and affects almost all basins and waters. The FAO reports that 28% of Panamanian soils suffer degradation, which represents a serious problem for the country's economy and development. This situation underscores the urgent need for greater investment in soil conservation and a more equitable distribution of wealth to ensure a sustainable future for all citizens of Panama.
Synthetic biology and genetic engineering have given us the opportunity to look at problems in different ways. As we have spoken with farmers and other experts in the area of environment and agriculture, we have improved our project to fulfill their needs.
Our solution consists of 3 genetic circuits composed of 2 chassis, the cyanobacteria Spirulina platensis and Synecocystis. The role of spirulina is the degradation of organic matter to produce humus, and that of Synecocystis is to produce a protein that can degrade chyolinitic clays in the soil.
We present to you our genetic circuit used for protein expression.
The first circuit will be used in the Spirulina platensis, and it consists of the following biological parts. First, we have the PDC1 promoter that regulates gene expression. We have a ribosomal binding site, or RBS, that facilitates gene translation. Next, we have our cell-5A gene, which encodes the cellulase enzyme responsible for breaking down raw material, and finally, the ADH1 terminator, that marks the end of genetic transcription, is coordinating the production of cellulase for the degradation of cellulose. The 3 last circuit will be used in the cyanobacterium Synecocystis. Our second circuit is composed of the constitutive promoter PGPDA that will regulate gene expression consecutively, a ribosomal binding site, or RBS, of course. We have also our NRPS gene, which encodes an enzyme that synthesizes siderophores using specific amino acids, and finally, the ADH1 terminator. The third circuit uses the same PGPDA promoter to regulate gene expression, which then passes to the RBS, following the COAA, COAD, ACCD and CARS1 genes, which participate in the synthesis of specific amino acids necessary for function number 2 and finalizing the terminator ADH1. And finally, we have a cell death circuit using the ICASP9 gene, which has the ability to induce cell death in a regulated manner through the interaction with GAL4 KRAB once the cell has fulfilled its specific
Mathematical
functions.
modeling has been one of the most challenging parts of our project in our country, since currently there is not much data that can be used for biological systems. While we spoke with different teams and individuals with knowledge on the subject, we developed a simple mathematical model to respond and analyze our project. The research project was multidisciplinary, involving a wide range of skills and knowledge. Our team was key in every stage of the process, from data collection to analysis and presentation of these results. To collect the data, we employ various techniques, from exhaustive search of reliable sources to the deployment of mapping technologies. The data we collected covers a wide range of variables, including nutrient content, texture, pH levels, and other essential indicators. Additionally, we thoroughly explored the ideal standards that should characterize soils in Panama, comparing the data collected with these standards to identify areas of improvement. Regarding the organisms Spirulina platensis and Synecocystis sp, our focus was on Spirulina platensis. We obtained detailed information about its cultivation, including the optimal conditions that favor its growth and production. We also investigate its potential application in the recovery of degraded soils. In addition to data collection and analysis, our team was responsible for creating advanced mathematical models that allow us to simulate different scenarios and evaluate the impact of introduction of Spirulina platensis on improving soils quality in Panama. This comprehensive approach allows us to not only gather valuable information, but also understand how this information can be translated into practical solutions to soil-related challenges in Panama.
As you may have noticed, our project depends largely on how farmers use the land. Listening to our target audience has helped us have a better project that can generate impact in our work area.
We also carried out different activities to engage with the new generation to educate them and invite them to the world of conservation and synthetic biology. Some of these activities include surveys, interviews, Instagram posts, collaborations, a large-scale event on this day to encourage both young people and adults that our soils need us. Human practices is an ethical and socially responsible approach to scientific research. In our iGEM EVOLUTECH Panama team, we address soil erosion, a serious problem in our region. We carry out various activities to raise public awareness about the issue in our project. Project 1. Soil erosion survey. With the help of the math modeling team, we conducted a survey of teenagers in Panama to assess their knowledge about soil erosion. Project 2. Expert interviews. We conducted interviews with professors specialized in remote settings to educate the general public about soil erosions. Project 3. Soil erosion brochure and team presentation. We create an informative brochure that addresses the problem of soil erosion in Panama and introduces our iGEM EVOLUTECH Panama team and details about our project. A science fair called GIS Day was held at the Agustiniano Chitre School, where we had a stand to talk about our project and hand out these brochures. This effort allowed us not to only gather valuable information, but also understand how this information can be translated into practical solutions to soil-related challenges in Panama.
Our project is not just a dream. We plan to make it a reality. We have already thought about the necessary steps to make this project a reality, designing all the experiments we want to do to further develop this idea. Here we will show you a little about our experimental design proposal. Our Spirulina platensis cultivation project, an an algae with great potential for food and the environment. To grow this algae in Panama, we chose a conical tubular photobioreactor, which takes advantage of natural sunlight and creates an optimal environment for the growth of spirulina. This photobioreactor has several advantages over other models, such as greater light collection, lower risk of contamination, greater control of culture parameters, and greater productivity. In addition, we use a drip irrigation system, which minimizes water use and protects the spirulina from direct solar radiation. With this design and operation, we seek a sustainable cultivation of spirulina that addresses global challenges related to food and the environment. Our team considered using the strains Arthrospira spirulina platensis NIES 39 and Synecocystis sp PCC 6803, which are non-hazardous to the equipment, the public, and the environment. We grow our organisms in a photobioreactor, where we regulate and monitor the biological conditions for the growth. These strains are known to not produce significant levels of cyanotoxins. Our project uses Spirulina platensis and Synecocystis sp to solubilize kalinitic clays, improving fertility and reducing soil erosion. The objective is the application of genetically modified organisms in agriculture to increase sustainability and productivity. We identify the biological, genetic, and inappropriate use risks of our project, evaluating their severity and probability of occurrence. We implement management and mitigation measures to prevent or reduce these risks, ensuring a safe and responsible project. Our team dedicated itself to developing a sustainable business. We carried out studies to identify the target market and market trends in biosafety. Our core values are sustainability, innovation, and social commitment. We have developed an action plan that includes the development of a prototype, market testing, obtaining financing, and launching it to the market. Our planning work has allowed us to identify opportunities and establish a framework for the implementation of the venture. We are committed to creating solutions that address global challenges related to food and the environment.
GROUNDSAVIORS is a forward-thinking project with many possibilities. Our project will continue to grow and improve as we acquire more information and feedback. We want to have a great impact in our community and even our country by carrying out more activities related by SynBio inspired by iGEM projects to carry out high-impact projects in our community.
Our team is incredibly grateful to everyone who helped on this project throughout this year of hard work. We will continue working to these kind of problems, and many more can be solved with the synthetic biology. We would like to thank iGEM Design League to bringing us together and making this opportunity possible.
We are iGEM, EVOLUTECH
Panama, and this is
GROUNDSAVIOR.